Well, hello everybody. First of all, I would like to thank uh, you for your kind invitation. First, let me introduce myself. I'm Dr. Manos Vlasiou. I'm a chemist working at the pharmacy program of the Department of Life and Health Sciences at the University of Nicosia in Cyprus. I'm teaching there lessons of physical chemistry, pharmaceutical chemistry, while conducting research in computer-aided drug discovery, mainly on metal-based anti-cancer drugs. Since cisplatin was applied as drug in cancer, chemists have tried to synthesize new alternative metal complexes as anti-cancer drug targeting to increase efficiency and lower toxicity. Numerous metals complexes with various ligands has been evaluated against cancer models. However, the majority of the anti-cancer drugs are non-selective and often lose their efficiency because of the constant mutation changes of malignant cells. So, the main advantage here of metal-based drugs over organic fabricated drugs is their mastery to alter the coordination number, geometry, and redox status, usually controlling their biological activity. Zinc and vanadium ions are essential to human diet, and they have been found in the active center of several enzymes. In particular, zinc is involved in several biological processes, and its deficiency can cause multiple neurological issues. Several drug compounds can form complexes with zinc ion during their transportation with human serum albumin. Additionally, we know that zinc complexes were evaluated for their anti-cancer properties and for their antimicrobial activity. Administration of vanadium compounds to humans have shown important biological response, including anti-diabetic and anti-cancer activity, DNA interaction, and reactive oxygen species formation, which appear to represent the main modes of action of anti-cancer active vanadium compounds. The relatively low toxicity of vanadium metal and the different oxidation states that it can occur is responsible for its extensive use by the researchers. Phenol redox active ligands and especially hydroquinones are key molecules in several redox biological processes. So, hydroquinone derivatives are participating in autophagy and apoptosis by Ross generation and also inhibiting tumor growth. On the other hand, hydroquinone ligation to metal ions is rare mainly due to the absence of coordination site and low pK values of the hydroquinone hydroxyl oxygen donor atoms. So a strategy to overcome this barrier, as you can see in this slide, is to synthesis of substitute hydroquinones in ortho position with substituents containing one or more donor atoms, thus enabling the metal atom to form halide rings. The hydroquinone derivatives were synthesized here from the corresponding tert-butyl or methylbenzene diol by following the classical conditions of the Mannich reaction for phenolic compounds. Okay, here you can see the synthetic routes for the main ligands that we created. More insights regarding the structures of the complexes after the complexation reaction of the ligands with metal ions of zinc and vanadium were given by density functional theory computational studies. We found there that the zinc complexes C1 and C2 are 5 coordinated, having a trigonal planar geometry, while the vanadium complexes C3 and C4 are having a more stable octahedral geometry. The 4 new complexes, we are talking about 2 new synthesized ligands. So we have 2 zinc complexes and 2 vanadium complexes, actually vanadium 5 complexes. So we have 4. The 4 new complexes were evaluated for their antioxidant biological activity. In our efforts to answer the question if our molecules have anti-cancer activity based on their pro-oxidant or antioxidant activity, we have determined the percentage inhibition on the DPPH radical. The radical scavenging activity values of the complexes are shown here, and as we can see, C2 at the lowest concentration has free radical scavenging activity. As the concentration increases, the scavenging activity decreases as well. The same seems to be true for complexes C1 and C3. So these molecules will have pro-oxidant activities, since at higher concentrations they cannot have free radical scavenging activities. On the other hand, the complex number 4, as the concentration increases, scavenging activity increases as well. Some compounds, though, form free radicals as a result of metabolism. So, therefore, with the DPPH study, which is cell-free, it cannot be determined if the actual mechanism of action is antioxidant. It is likely that in vitro in cell system, the compounds show pro-oxidant or oxidant activity, which needs to be determined in further studies. The absence of a cell in vitro system cannot provide further information. The covalent attachment of organic intercalators to transition metacomplexes can lead to novel DNA interaction that affects the biological activity. These metacomplexes have R-bonded aromatic sidearms and can act actually as a dual function complexes that bind to DNA both by metal coordination and through intercalation of the attached aromatic ligand. To compare the strength of the binding with the DNA, we provided measures after DNA interactions with UV visible studies. We saw that C4 molecule binds stronger with DNA base pairs, followed by complex 1, 3, and 2. Additionally, as you can see in the slide, viscosity measurements were made with increasing amounts of complex to DNA solution. Normally, when the complex is binding with DNA base pairs, it will be lengthening and stiffening the DNA double helix. So this is going to lead into increased viscosity of the solution. So with increasing amount of complex, the relative viscosity of all DNA solutions increased. The viscosity was most pronounced when interacting with the zinc complexes, while the lowest viscosity was measured with the vanadium complexes. So here you can see that the zinc complexes interact stronger with DNA base pairs. Therefore, we can say that the zinc complexes are favorable to act to the DNA, while the vanadium complexes in a different area of the cell. So this is one remarkable result. Let's move forward. We have here further biological studies with the bovine serum albumin. We provided here studies with the UV. So as you can see, strong absorption peaks was observed in 259 nanometers and 370 nanometers, and the peak increases with the concentration of the 4 complexes. So furthermore, the formation of C1 and C4 bovine serum albumin complexes resulted a slight shift from 259 to 249 of the spectrum towards longer wavelengths, indicating that the interaction between complexes and bovine serum albumin is quite strong. Similar activities were indicated for the complexes C2, 3, and 4, and also the cytotoxic properties of C1, C2, C3, and C4 complexes against the breast cancer cell line MDA-MB231 can be seen here as well. We provided MTT assays here for the determination of the antiproliferative effects of the prepared zinc and vanadium complexes compared with the well-known metal-based anti-cancer drug cisplatin. As we can observe, the first complex of vanadium has the highest cytotoxic activity, while at all concentrations the breast cancer cells died, followed by the second complex of vanadium. Also, vanadium complex had the highest activity at lower doses, even compared with cisplatin. Here it is worth to mention that these cancer cells, these breast cancer cells, were resistant to cisplatin, and our vanadium complexes were able to kill these cisplatin resistant cancer cells. Okay, what we have here more, actually we managed to perform some antibacterial studies in order to see the antibacterial activity of these complexes. Actually, we compared the activity with the antibiotic chloramphenicol, and we saw that despite the promising anti-cancer results that we observed by the molecules, the antibacterial activity results weren't so good. Only the second complex of zinc showed adequate activity against E. coli, which was the representative of gram-negative bacteria, while we had also a representative of gram-positive bacteria, which was the Staphylococcus aureus.